Namaste students. I am Jevin Dr. Aziz Mahmood Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the topic antibody structure, and this topic belongs to the principles of immunology, immunotechnology in the Department of Food and Biotechnology students. So antibodies, what are the antibodies? In the previous video, we have discussed about antigen structure, complement system, innate immunity, adaptive immunity. So antibodies are part of the adaptive immunity. That is the third line of defense system. And what is the source of these antibodies? Antibodies are secreted by the B cells. When the B cells gets activated, they differentiate into the plasma cells and secretes millions of antibodies. Millions of antibodies are secreted by these B cell and plasma cells. B cells which secretes antibodies, and then after secretion, antibodies have important function. These antibodies are specific for a particular kind of antigen. They bind to a particular kind of antigen, and after binding, they identify antigen and help the immune system for the elimination and the killing of that particular kind of antigen. So this is all about the normal introduction of the antibodies. What are antibodies? And in the detail, we are going to discuss that what are the antibodies and what is their functioning. Antibodies are generally some protein structures. They are protein which are secreted by the B cell, and they are prepared according to the epitopes which are present in a particular kind of antigen. A antigen has a specific type of epitope, then to that kind of epitope, a particular kind of antibodies will be prepared and it will be secreted. So now we'll talk about the structure of the antibody. Antibodies are made up of two light chain and the two heavy chain. The light chain are identical to each other and the heavy chains are identical to each other, means they are very similar. The size of light chain is smaller, that's why they are called it as light chain, and the size of the heavy chain are larger, that's why they are called it a heavy chain. The size of light chain is 25 kilodalton, and the size of the each heavy chain is 50 kilodalton. Light chain and heavy chain are bound to each other by disulfide bonded. Means a heavy chain and the light chains are bound to each other by some specific bonds which are known in the disulfide bonds. And each heavy chain is bound to another heavy chain by hydrogen bonds, H bond. 100 amino acid terminal vary of both heavy and light chains. If we talk about the amino acids, then the amino acids of the heavy chains and the amino acids of the light chains vary to each other. And they also vary from one antibody to another antibody. And there are some different domains also for antibodies, which are referred to as a VL variable region, VH heavy region, CH constant heavy chain region, CL constant light chain, and there is also a special region which is known as CDR region, complementary determining region, which bind to the antigen CDR region, and remaining regions are very similar to another class also. The antibody have approximately 110 amino acid sequence and interchange disulfide bonds with each other. If we talk about the heavy chain has a one heavy chain at the three or four heavy chains may be there. For example, if we talk about the constant region, in the constant region, there are two regions. One is variable region and the one is the constant region. Variable region is the region where the region the antibody differs from each antibody to another antibody. Means this region will determine the type of antigen to which it will bind, and the constant region will be same in all the, and there may be different concentration. For example, CH1, CH2, CH3, CH4. It depends on the size of the antibodies. And in the light chain, there are some one one variable region and one constant region, that is the VL and the CL. Hinge region is the region where the light chain and a heavy chain will bind to each other, and hinge region is rich in the proline. It makes the antibody in a flexible form, and hinge region is found in the IgG, IgA, IgD, which are the immunoglobulins. And we will discuss the type of immunoglobulins in the next video. And proline residues are target for proteolytic digestion, and they are rich in the cysteine residues. IgM and IgG lack hinge region; they do not have any kind of hinge region. And IgM and Ig and they do not have any kind of hinge region. So, if we talk about the enzymatic digestion of the immunoglobulin, and if we digest the immunoglobulin, how we came into know that immunoglobulin have heavy chain, light chains? What is the basic idea? The basic idea is that if we digested the antibodies by some enzymes, then we came to know that these are the particular areas of an antibody. So if we digested the antibodies with the papain, then it generated three fragments, two identical FAB portion and one FC portion. FAB portion is also a part of the antibody. And what is the FAB portion? FAB portion is the portion where antibody binds to the antigen binding domain. And FC region is the region where the antibody binds to the cell surface. And papain digestion makes some FAB and FC region. What is the sequencing of heavy chains? Sequencing of several immunoglobulins reveal that the immunoglobulins have 100 to 110 amino acids, which are highly variable. Five basic sequences are there: alpha, beta, delta, epsilon, and mu, which generate different kinds of antibodies, such as alpha will generate immunoglobulin A, gamma will generate immunoglobulin G, delta will generate immunoglobulin D, epsilon will generate immunoglobulin E, and mu will generate immunoglobulin M. So these are the heavy chains which are present. And on the basis of these heavy chains, five types of immunoglobulins are there. And these class are known as the isotypes. IgA, IgG, IgM, IgT, IgE are known as the isotypes. Each class can have any kind of light chains, kappa and lambda. So there are five heavy chains: alpha, gamma, delta, epsilon, and mu. And when we talk about the light chain, there are any kind of light chain, can be kappa or lambda. Two light chains are there, kappa and lambda light chains. And the difference from one antibody to another antibody is on the basis of the heavy chain. And on that basis, antibodies are divided. And CDR regimen is also a hyper variable. CDR1, CDR2, CDR3, they may be vary. And on those basis, we can identify a particular kind of antibodies. So now we must understand the structure of antibody. So this is the structure of an antibody. Here there are uh, domains. This domain is known as there are two chains, light chain and heavy chains. So this is the heavy chain and it is light chain. In this region, there are some regions which are constant region and some regions are variable. The variability is found in this region here, from this here, from here. So variability is found here, not anywhere. And from here to here, the region is constant. There is no variability. So it is known as the constant, and and this region which vary is known as the variable region. Heavy chain has a variable region, and that chain also has a variable region. Yes, VH means variable region of heavy chain, and VL means variable region of light chain. CH means heavy constant region of the heavy chain, and Cl means constant region of the light chain. They are bound to each other by disulfide linkage. Okay? They are bound to each other by disulfide linkages. Yeah. So this region is the region which will bind to antigen, and it is known as the antigen binding domain. This region is region where antigen will bind, okay, at this region, and this region will differ from antibody to antibody. And this region is the region which will bind to the cell. Okay, this region is present on the surface of the cell, so it is known as the FC portion. So VH and VL are the variable region of heavy chain and the light chain. CH1, CH2, CH3 are the constant region of the heavy chain. The light chain is also there. The two types of light chain will be there, kappa and lambda. And heavy chains, if you talk about the heavy chains, there are five heavy chains: mu, gamma, alpha, delta, or epsilon. The region, this is the hinge region which is present between the heavy chain and the light chain, and this is the carboxyl group, and this is the nitrogen group. amino group which is present on the surface. The two ends are there, amino group and the carboxyl group, because this is a amino acid also, and this is a protein. So in some more detail, here are some disulfide linkages. This is the light chain, this is the heavy chain, and this is the FAB portion here. And if we digest it from a papain, then we get the portion which is not the as the FAB portion and the FC portion, and also we get the heavy chain and the light chain. More deeply, when we talk about the structure, this is the structure which is known as the normal structure, and this structure belongs to the alpha delta heavy chain. This is the variable region of the light chain. This is the variable region of the heavy chain. This is the constant region of the light chain. This is the constant region of the heavy chain. And this at this region, the antigen will bind. So this is known as the antigen binding domain. This all this area is known as the antigen binding domain. On this area is known as the FC portion. So this is all about the structure of antibodies. In the next video, we are going to discuss about the type of uh, antibodies and their functions. Thank you. So, which kind of questions can be asked in this antibody structure? So, the examiner can ask that what are antibodies and explain their structure. Explain the heavy chain and the light chain. Explain the components of the antibodies. So, everything can be asked in the lecture. So, my lecture is over here. Thank you very much.